We'll just get started. That's okay, everyone? Now? All right. So we might just get started if that's okay with everyone. So good morning. Thank you for coming out this early. I know it's not the easiest on a Saturday morning. I'm Hemant Nelvagal, and I'm with my colleague Keigo Takahashi. And we are researchers working in the lab of John Cooper at Washington University in St. Louis. And we're running a session this morning that's Genetics 101. And basically the point of this session, as I'm sure some of you have been to these before, and for those who haven't, the point of the session is sort of like just to level the playing field. So everybody has different amounts of how much they know, myself included. Sometimes I don't know stuff. So this is just basically to get everyone kind of oriented before we go into the science talks, and also maybe break down some of that jargon that us scientists love to use, all those big fancy words, and just try and give you an idea of what that actually all means by the end of it. So without further ado, to get started, right, so who's heard of cells? Everyone, great. Okay, so we're all made up of cells, all of us. Everything we have is cells. And even before we're born, when we're conceived, we start off as a single cell. And that single cell becomes a multiple of cells, and that just keeps growing and growing and growing. But as we can all tell, I'm pretty ball-shaped, but we're all not just balls of the same cells, right? So I might be. So those cells differentiate, and they become different types of cells, and you have muscle cells and skin cells and liver cells and all of that. But somewhere along that line, from going from a single cell to this complex organism that we call a human being, there must be some instructions somewhere that tell these cells to become all these different types of things. And we as scientists, part of our job is to understand how that happens and why that happens and so on and so forth. So basically, those instructions and understanding how that translates is genetics, right? So just to get started, a cell can take many, many shapes, and depending on the shapes, they call it a bunch of different things. But just a general cell, we can draw this little ball, right? And oh, sorry. I just run around it. Is that better? Yes. Okay. So I don't really crowd Keigo out. Right. So the cell is made up of a bunch of molecules. And everything that's, for the most part, in a cell is made up of these things called proteins, right? And that's just the word we use for them. And basically, everybody's kind of heard it through high school. Proteins are the building blocks of the cell, and amino acids, right? So basically, everything in the cell is made up of these things called proteins. But how do proteins get made? And different cells have different quantities of different proteins, right? And there's like a million types of proteins. What tells a cell to make them? So in a cell, there's usually a compartment inside called the nucleus, right? And the nucleus contains a type of chemical, a type of molecule that I'm sure all of you have heard of. It's usually written as an acronym. It's called DNA. So everyone's heard of DNA, right? So anyone can tell me what DNA stands for? Not John. Cheating. Close. Yes. So again, fancy chemical terms, right? So it's deoxy, right? Ribonucleic acid. And I'm not really concerned about this because, like I said, it's all just fancy chemistry terms, right? But basically, that is the recipe book, or at least the language in which the recipe book is written to make all these proteins, right? So the cell goes into this and reads particular parts of that recipe book and makes the different different proteins that then make the cell. So it's a self-contained mechanism. So to tell you more about how that works and how DNA is organized, I'm going to hand over to Keigo, and he's going to take it from here.
Good morning, everyone. As Hemas explained, my name is Keigo. I'm a graduate student at Dr. John Cooper's lab. So Hemas already explained that DNA, basically gene is written in DNA. So I'm gonna tell you a little bit more details about DNA. So if you look at the cells using microscope and then really zoom in, you can see that there is inside the nucleus, there is some structure which looks like this X shape, like this. So this structure is called chromosome. So basically, DNA is kind of integrated or like packed into the chromosome. So if you really more like zoom in, like for example, this here, and then you can see some like this double helix structure. I think it sounds kind of familiar to everybody. And then, so DNA is basically made up by 4 letters, 4 alphabets. This is like A, T, G, C. Then if you really closely look at this double helix, you can tell that okay, so here is A, here is T, here is G, here is C. And basically the lineup of these alphabets is gonna be information about gene and how to make the protein. That's basically the concept of DNA. Then, so let's go back to the chromosome. So does anybody know how many chromosomes humans have? I'm sorry? 23? Pair. I got some correct answers. So it's true. There is 23 copies of chromosomes. So basically, in total, humans have 46 chromosomes. And then sometimes people get confused between gene and chromosome because there's disease which caused by the problem of this level, this alphabet level. Also, there's disease which caused by a problem of this chromosome level. And then the problem is sometimes both disease from both conditions are called genetic disease at the same time. So it's kind of confusing. For example, have you ever heard about Down syndrome? Or, okay, I think it's very famous genetic condition. People say it's genetic disease, but actually the Down syndrome is caused by one additional chromosome, actually chromosome number 21. So patient has one extra chromosome number 21. So this is just an example of chromosome disease. And then some people may say, so when people just, if you never heard about this concept of chromosome or genes, you may kind of mix up between chromosome disease or genetic disease. Actually, Batten disease is caused by the problem at this level, so gene level. So then I'm gonna explain more details about it later. So basically, I just wanted to explain that gene is integrated into chromosome. Then chromosome locates in the nucleus, and then this gene is again, as Hemas told, gene is used as a recipe book for protein. Okay, so then most importantly, so far, do you have any questions? Is there something unclear? Nope. Okay, so okay, let's keep going. So as I said, human has 23 copies of chromosome. That means half of chromosomes are coming from mother and half of the chromosome are coming from father. So that means all genes in our body have two copies. One copy is from father, one copy from mother. This is a way to transmit the genetic information from both parents to next generation. Then this process is called inheritance. Then it's very important to understand the concept of inheritance because it is very understanding the concept of inheritance is essential to understand how genetic mutation causes Batten disease. So I'm gonna just go over the basic concept of inheritance. So, as I told you, all human has two copies of gene from both mom side and dad side. Okay, let's say this is dad, this is mom, and then dad has two copies. D1, I said D2. Mom has mom M1 and M2. And then when parents make a next generation, there are equal probabilities for each copy to be transmitted to the next generation. So if I draw the diagram, this one is D1, M1, D1, I'm sorry. Yeah. M2, D2, and 1, D2, 2. Right? So there are only 4 combinations. And then there's equal chance for each combinations to happen. So if you calculate the percentage, each of them have 25% of chance. So this is very basic concept of inheritance. So then my question is, in reality, these two copies are not necessarily same. Then the question is, which copy of gene actually results in the characteristic of the body? Okay, so let's say, for example, there's a gene which determines the eye color, right? And then I'm gonna, for example, okay, I'm gonna very simplify it. For eye color gene, there is one gene which confirms this brown color. And then this is capital B. Then this small b confers the blue color for the eye color, okay? And then in order to understand this concept, I have to explain the word is dominant or recessive. So capital B, which is brown color, we know that brown color is dominant. And blue color is recessive. So what does it mean? So basically, brown color is dominant to the blue color. That means brown colors is preferred to blue color, or in other words, brown color can mask or overwrite the effect of blue color gene. So, in that case, if you have one brown color copy and one blue color copy, so you're going to have brown color eye. It's going to be brown. Because, as I told you, brown color is dominant. And then only if both of copies are like blue, blue, there's no brown, right? Only in this case, you're going to have blue color eye. So this is important to understand this concept first, because this concept also applies to the disease, the mutation associated with Batten disease. Okay, so I'm gonna draw the same diagram. So let's say both of your mom and dad have a, that's okay. One copy of brown color, one copy of blue color. And then when they make a child, there is an equal chance of yeah, these are 4 combinations that could happen. And then, if the child received this brown and brown, the eye color of the child is going to be brown. And it's the same for here, because brown is dominant to blue. So these are also going to be brown color. The only if the child has 2 blue color copies, then child is going to have blue color eye. So the percentage of child got a blue color is going to be 25%. So why I explain this is because this concept of dominant or recessive it also applies to the inheritance of disease mutation. Then in almost all cases, Batten disease are recessive disease. That means it's only if we have 2 mutations in the same gene, then only in this case, you're going to have a disease. So in that case, so this is how the recessive disease will happen. So the important thing is, so when we do the genetic test, what we are going to see, what genetic tests reveal is that there are 2 things. A, how many disease mutations that each person have. 2, what kind of mutation they have. Yeah. So now I'm going to talk a little more details about what is mutation. Okay. So do you have any questions so far? Okay. Okay. Cool.
All right. So everybody's okay with how things are inherited and what's dominant and what's recessive. Yeah? So everybody's also heard the term, I'm sure, mutation. thrown around, right? It doesn't always mean you become an X-Man, I found out, but you do get superpowers. So a mutation is basically changes that happen in the DNA, right? So the DNA is like a language of its own with just 4 letters, and it, as Keigo already mentioned, it's got 4 letters, and these are written in various combinations. Now, as again Keigo mentioned, bits of this DNA form together to form a gene, right? And each gene then makes a protein. So now, each one of these letters is important to make sure that the protein is made the right way. Now, if there are changes in that, that's what we call mutations. Now, mutations can be changes to this in multiple ways. They can be what we call deletions, where one of these letters is removed. It can be insertions, where a new letter might be added in, and might be substitutions, where instead of one of the letters where that's supposed to be, another letter takes its place. Yeah? And we can go into details about how these mutations happen. Sometimes they're just one letter at a time, like the examples I gave you. Sometimes they're whole groups of letters, and sometimes they're whole chunks of the gene as well, right? And so you can easily tell that depending on the kind of mutations you have, even within the same gene, it can lead to different results. Yes? Okay, so, sorry, it gets a bit messy. So the end result of mutations are different. Everybody has mutations. That's how we all ended up being different in the first place. Everybody that's born has mutations. Only when these mutations affect the genes that are responsible for proteins that are functional and change the function of the protein do they start to cause disease. Now, mutations causing disease can happen in 2 ways. Either one, the changes in the mutation lead to there being no protein, right? So the mistakes happen and the machinery that's reading the DNA goes, all right, that's all wrong, we're not going to make any protein anymore. Alternatively, you might get some form of protein that functions to a different extent than it's meant to. It might function at 50% or 75% or 25%. And so even just depending on that mutation and the kind of change that you see, you might get differences in what we get in terms of protein expression. And this is what Keiko will talk more about.
Okay, so this is a basic concept of why mutation caused either no function or like less function of protein. And then, so let's go back to the Batten disease. So Batten disease is caused by a mutation, which everybody knows. Then the mutation typically happens to the gene whose product is associated with one specific structure of the cells, which is called lysosome. So, as many of you know, many of you may know that lysosome is basically a recycled being in the cells, because cells have to cell use the proteins, and then there's so many proteins the cells have to produce. So what cells usually do is after making proteins, and then cells is going to recycle the proteins. So protein is like a kind of made up by a lot of like a Lego box, and then so when cells recycle the protein, so lysosome, so each oral protein is going to be incorporated into the lysosome, and then the lysosome is going to basically degrade or kind of break up each proteins into each piece of Lego. Then, after breaking up this Lego is going to be kind of secreted inside outside the cells, and then the broken Lego is going to then recycled to make different kind of proteins. So basically, there are two kinds of proteins. There are two kinds of proteins in lysosome. So one is the proteins which it's kind of like scissors, which gonna break up the proteins. So this is called enzyme. So enzymes actually play a major role to just break up the proteins. And there's another kind of lysosome protein, which is located at the membrane of the lysosome. So membrane is just kind of surface of the lysosome. Then having a nice wall of the lysosome is important because if there is no wall of the lysosome, this enzyme can go up everywhere inside the cells, and then it's going to basically break up everything. So having both appropriate enzymes and membrane protein are very important to maintain the function of a lysosome. Then, Batten disease are caused by mutation in gene, which makes both enzyme and membrane. So actually, enzyme or membrane. So then, this is kind of confusing point because when people say here just lysosome disease, people don't necessarily understand there's two different kinds of proteins in lysosome. So and then why it's important, the reason why important to understand the causes of the Batten disease is either enzyme or membrane is because it's going to change their strategy for the therapy. So Hemant is going to finish up about this, how the difference between this membrane or enzyme is going to affect the therapeutic strategy.
Excellent, thanks. So again, at this point, we can jump off and say, is everybody clear on that? Any questions about what the lysosome is, where it is in the cell, and what it does? Or any general question? Yes. So this is generalized for all forms of Batten disease, right? And the idea is, and I'm sure you've heard the term lysosome thrown around, lysosomal disease, right? And the point is, lysosomes are these bits in the cell that break down stuff, as Keigo explained. And different mutations in different bits, so every bit of the lysosome, whether it's a protein in the wall or the membrane here, or whether it's one of these little scissors that breaks up stuff, is coded for by a single gene, respectively. And depending on which gene is mutated, you get defects in that respective protein. Now, all the proteins are in the same place. They all do different functions. So each of these scissors breaks down a different bit or a different connection. And every bit in this membrane performs a different function as well. And so depending on which of those proteins is mutated, you get different forms of disease. Yeah? And so, yes, and that's a very good point. Thank you for bringing that up. There are some types where we don't know what the protein's function is, and sometimes there are mutations of unknown significance where we sometimes don't even know what the protein itself is. And that's a very important thing to highlight here is this is an ever-growing field. It isn't like a textbook that's written and published once and it's done. We keep adding to that repository of knowledge as we try and do it. That's kind of our jobs, right? So as we find out things, like for example, even with some of the Batten disease, we know what the gene is, we even sometimes know what the protein is, but do we know what that protein does? Not all the time. Yeah? And yes. Absolutely. Yes. But we do know where it is in the cell and what we think is being affected. So the other term that we throw around quite often is not just lysosomal disease, but lysosomal storage disease. Have people heard that? So lysosomal storage disease, right? So that storage is an important concept. So like we said, these things here break things down. So it's like having a recycling bin in your house, just as Keigo said. So if things are not being broken down on time, they just keep building up, right? And that's what we call storage material in the cell. So all this unwanted stuff keeps either building up inside the lysosome or outside it, depending on which proteins are affected, right? And that is why it's called lysosomal storage disease, right? So the strategies to treat it change depending on which bits are mutated, okay? Now, these scissors are quite interesting because we consider them soluble. So you put them in a solution, they dissolve, right? So what's interesting about them is cells produce them, and then they are put into these lysosomes. Equally, when a cell makes this, sometimes those will be secreted outside the cell. And that can be taken up by other cells because they have specific sort of gates that allow for them to be taken up by other cells. Yeah? And so if none of the cells are producing it, and if we give these proteins, these enzymes, into this space, they will be taken up by the cells, right? And all of you are familiar with, enzyme replacement therapy. You've heard of it, I'm sure, right? And that's exactly what it is. We are providing that missing bit of enzyme, if it's soluble, that can be taken up. Now, that's only for the bits that are soluble. If you have things that are stuck in the membrane or the wall of this lysosome, those are not secreted. Yeah? And that's why it's important to know which is which, right? And then we have to come up with different strategies to try and make this, replace that there. You can't just give it and it won't be taken up. It'll just stay here, right? Okay. Yes? That's a great question. Lysosomes are in every cell in the body, okay? But lysosomes, like everything else in the cell, is kind of specialized in each cell. And the functions they do are specialized. And we're almost just scratching the surface of what lysosomes actually do. To say that they're recycling bin is nice for a conversation like this, where we can sit and say, okay, they're breaking things down. But the truth is, it's far more complicated than that, and they're doing a bunch of other things just for regular functioning of the cell. And so, depending on which protein is affected and whether that affects that particular function of the lysosome, different cell types can be affected. Now, in Batten disease, that usually means, historically, that the nervous system is the most affected. But there are other lysosomal diseases which spare the brain entirely. Yeah? Any other question? Right, with that, I'm getting the sign. I'll say thank you, and well, if there are any other questions, come and find us. Yeah?
Thank you so much.
Thank you.